Hi, this is Selim Ünlü. I have some technology questions. What do you expect? So one for Michael. So the large field of view, high resolution imaging with the synthetic aperture, what is the numerical aperture of the lens? Because one of the problems is that you're going to lose your light collection efficiency. That's going to be the contrast of the fluorescence imaging if you use a large field of view, small magnification, and NA is also small. But that's the illumination.
Okay.
You are illuminating at different angles, but collection is through.
The collection is essentially identical to any other scope that we use. The same exposures. I mean, it's not a long exposure at all. It's still millisecond exposures.
Okay, perhaps I misunderstood the technique.
Okay, I have some questions from online. For Vasilis, and this is one of our questions that we had thought about beforehand as well, is how do you quantitate variability between cells in single-cell analyses, and how does the variability compare with when you look single-cell analyses compared to one another, compared to doing bulk cell analyses?
This is actually a very good question. This is one of the problems we have in terms of the variability and between different cells. So most of the signals you're getting based on intensity. So this is one of the weak areas we're trying to work on it and trying to minimize the noise and the variability. Now, you have a number of steps that you can take in order to try to avoid that. So especially when you do the single cell imaging, you have to use an automated sprayer in order to avoid, to avoid differential matrix coverage, which will result to different ions that are molecules that have been ionized. So, yes, we recognize this might be a problem. We try to minimize that as much as we can. And the automated sprayer, or even if you go on a single cell, if you have a robotic system that will minimize the variability.
So I just want to clarify one thing, which is when we talk about variability in this context, there is a technical variability of ability to measure or whatever influences or the way we measure, versus the variation, if you will, of cell to cell variability. And those are orders of magnitude different in some cases. So the technical variation, even if it is present, it may not affect, it's not going to yield the orders of magnitude cell biological variation that we see for sure. So there may be some room here to sort of live with a certain level of technical variability noise, if you will, and still make meaningful inferences.
Yeah, so my question is directed to Dr. Kaminski, but others obviously can chime in also. So you mentioned this a little bit in your talk, and that is the methodology for isolation of cells, and the thing that I think often worries investigators is as we manipulate these tissues, are we changing the gene expression profile in these preparations? And we can always say, well, we'll compare it to control because we're isolating the same way. But in reality, diseased tissue or tissue perturbed by xenobiotics may respond differently to the way we handle those tissues, even if we handle controls and the affected tissues the same way. So I'm wondering, as you think this through, what kinds of controls can we include to try to convince ourselves that the changes we're seeing aren't just due, or at least in part, to the way we're manipulating these tissues, especially with the sensitivity that we now have to measure, for example, the transcriptome.
Frankly, there's no way we can really fully convince ourselves that what we're seeing is not a result of the tool we're measuring. It's actually equal, like I see you better without my glasses now. Right. So I think it's very similar.
Yeah.
And I think what we need to agree is on set of parameters we record that are shared in the community, which is much better. So, for instance, and this has to do with many years ago, when I had a warm autopsy project for obtaining lungs from dead donors, we initially thought that we should get the lungs as quickly as possible. What happened was that we actually increased variability because the difference between 30 minutes and 60 minutes of warm expectancy is double. Whereas when we said we're going to do all the lungs within 6 hours, then the difference of 20 minutes became very small. So I think that coming up with very pragmatic approaches per tissue is going to be important. And recognizing that there's not one good. So you have to replicate your data, you have to triangulate. The reason we're really so convinced about this data is actually that it really recapitulates our bulk data. There's new things, but actually the sanity check makes a lot of sense. I am a little bit concerned with the fact that we don't have a good, in highly diverse gene expression, single-cell gene expression data, we actually don't have a really good way to measure or to really say specifically differential expression per distinct cell. I think that's where more complex statistical modeling is really required, because we're really good at identifying the cell groups and identifying it then, but I think there's some issues which are computational, but other things. But I'm less worried about it's just going to be altered. What you don't want to go is to the purest approaches, because I'm going to assure you that if you dissociate the lung, then run flow cytometry, then with 3 antibodies, then you definitely change gene expression.
Thank you.
I am Patrick McMullen from Cytovation. Toxicology right now is in the middle of this transition. We're trying to use fewer animals for assessing chemical safety questions and more in vitro and more in silico tests. The way I always envision that is you have some finite number of assays or test systems, based in cell lines or based in cell-free systems, and some finite number of endpoints that you're measuring. Is that insane to think that we could use a collection of systems like that and cover all of biology, or enough of biology to be able to make good decisions? Coming into this, most of you talked about variability, within a cell population, across cell populations within a tissue type, between individuals in the same tissue type. Is there hope for taking the complexity of that variability of those tissues and transitioning into test systems where we could measure something and be reasonably assured that a chemical is not going to be causing problems in people?
Anybody want to dive into the deep philosophy there?
I'm not sure you can answer that. But one thing is that I actually am really interested in the site of these more cellular circuitries in the context of an organ. Because my assumption is that for the milder injuries, we probably can handle most of them, right? It's just a matter of quantity and repeat. But for these milder injuries, when they just affect one cell type, usually, it's when you change the homeostatic nature of it. So maybe our approach should be a little bit more of integrative perturbations and actually look how you change the homeostatic nature of an organ or a model of an organ, a lung on a chip or whatever, instead of necessarily looking at the beast-to-beast cell like we've been doing. So I think that's where the sort of the systems type approaches could really help us.
Growing cells on plastic. Growing cells on plastic is not the greatest way to do anything, but we've been doing it for a long time. Everyone has.
Actually, it's worked.
The shift to more biological culture systems, it's ongoing all over, creates all sorts of new ideas, new problems, new challenges. I don't see any one thing going to solve anything. We're happy we can do what we do and reduce the number of experiments and different types of experiments instead of a bunch of disparate data and trying to figure out, we can say, well, we got it all from this model. Well, then we got to model the model, change the model and grow it. And that's ongoing, but it's not going to happen instantly.
If I may add to that, as fantastic as these methods are in giving us an extremely high resolution, possibly even high throughput view, it still doesn't take away the problem of mapping them back to a much lower dimensional, in a toxicology world, I suppose you would call it mode of action, for example, or mechanism in a basic science, or in a control engineering terms, critical control points where that you can manipulate and move. And that's where you got to derive a certain insight into to go do the follow-on and other types of experiment, rather than continue to be in a higher dimensional space. That reduction into that's how I see it. The reduction into a specific regulatory understanding of what drives the high resolution biology, if you will, is what would be needed.
So I was thinking, perhaps this is also a place where knowledge transfer, using artificial intelligence can help us to learn across different systems, to see what kind of functions, modules they're preserved, going from, like a simple organism like a cell culture system to three-dimensional cell culture system, and then to organoids, and then to PDX model, and then to human. So I feel like there's a hope, though. You have more controllability at the lower level, lower complexity level, so you can maybe can afford to scrub more and learn more. And then when you move up higher and higher, more precious samples, you give less of the, then you have more and more constraints. So maybe, some research effort on this direction would be very helpful.
Kim Buckelheide, Brown University. This is sort of a RAF off of Raja's picture of Waddington's, designation of a cell, and the channel gets really deep, and you're sort of stuck, with whoever you're going to be through a developmental sequence, as opposed to a landscape that has lots of dips in it and a lot of variability of cell types. And I think one of the messages I've heard so far today is that there are many, many different cell types that we used to lump together in our morphologic classification of tissues. And one of the things I'm struck by in the measurements, particularly single cell RNA-seq kind of measurements, is the static nature of those measurements and the potential dynamic nature of the cell changes that could occur. And so does anybody have any insight on the temporal nature of the ability to shift between these minima of cell type in a landscape? How rapidly that might be occurring? Because we're seeing lots of different cell types that are now more closely related. But we still have this construct in our heads that they're designated from birth.
So I can speak a bit to that rate at which the state transitions might occur. That really depends on the cell type and the context, if you will. In the liver regeneration context, for example, the macrophages have to shift their states in particular ways and start producing the right cytokines for the rest of the machineries to start getting in place. So that happens fairly rapidly. Relatively speaking, it doesn't take a day or two. It takes minutes to less than an hour for the state transition to start occurring. The cells entering cell cycle, obviously there's a binary decision at some level, and so that takes time, but there is a lead up to a gradient of cell state that walks up to that in some way, and that takes time. Because of accumulation of molecular entities, if you will, and waiting for some gatekeeper checkpoint signals to come through. So that takes time. So there is a full range of order of magnitude of cell state transitions that could occur.
So at least in sort of an area that's close to my interest, which is lung development, the work on new lineages of cells and how the differentiated is happening. There's already been new insights, but also real confirmations that some steps are really pre-designed. They happen, right? It's not just a result of us averaging everything. So a cell has to express, FOXJ1 that then needs to be activated to become a ciliated cell. So I think actually some of the reassuring, but the difference I think is, our problem with developmental biology and maybe disease was that we could never calculate the kappas. We could never say what is the rate of things. So I think we could translate biological systems into sets of equations eventually with single cell data, not necessarily now.
What would be most useful is to start accumulating evidence with live cell imaging, for example, that can be done from a morphological perspective and follow the same cell as it traverses states. And the correlations between the molecular and morphological are emerging now. So if you start accumulating knowledge that starts informing us which states are reachable, depending on where you are in the landscape at any given moment, what is that nature of that flexibility and the constraints? Is there a sort of a average path that most of the cells would have to go through this landscape, through the valleys and the hills? Is the path alteration is what the disease does, or the valley hill alteration, is that what the disease does? And they're fundamentally different things in terms of rewiring, right? So
maybe I could ask Dr. Biteen, could you comment a little bit about what you see in terms of the dynamics in terms of protein expression with the systems that you use in the single molecule analysis?
Sure. So it's interesting, right? Because all these dynamical scales are very different. And so when we think about cell differentiation, we're talking about hours. And when we think about subcellular motions, we're thinking about seconds. So bridging those gaps are really interesting. I think I'd mostly say I agree with what Raj was saying about using multimodal imaging in order to bridge the timescales. So at one end, we're limited with GFP and fluorescent protein labels that they take about 30 minutes to mature. And so any processes that really occur faster than that in terms of expression, we're stuck. And so for those, you want to go to different modalities. We can look inside cells on the scale of minutes, and then to go with more single cell imaging rather than single molecule imaging could take you to hours and days. The other really interesting thing, I think, beyond the multi-time scale is a multi-size scale. And in particular, going from a single bacteria cell or a single mammalian cell out to tissue and organs is a really important challenge. So there's a lot of new advances in light microscopy that are helping with this. For example, light sheets to be able to optically section living tissues and different modalities in that way.
Yeah, hi, Catherine McClelland, NIH. This is sort of a general question that I think is the elephant in the room that Lana kind of talked about, but as we get more and more cells that we can do these technologies on, we notice more and more subtle differences between what we thought were fixed states at the end of sort of the Waddington path. But what we have not really been able to do is repeat ourselves because these technologies are so prohibitively expensive. So there's the cost of the computation and getting the people to do the analysis, but also in many of these situations, the cost of just running the initial assay, getting the cells, having an expert who can dissociate them appropriately. All these things mount up, so we're now at a point where we have a lot of information, but not a lot of repetition to show that this is the ground truth. So I would ask, what do you think we can do to move into a situation where it's more egalitarian for any lab to be able to do these technologies, but also so that we can sort of fact check ourselves with the data we've already created and the models we've construed from that?
That's a very good point. I need to remind you, when the Human Genome Project started, the cost of whole genome sequencing was really big. Now it's
We're in the
down.
Wild West.
Huh?
We're in the Wild West at the moment.
But what I'm saying is, first of all, you need to increase reproducibility on your data. I completely agree with you. And there is a lot of variability. So developing enough deep learning and develop good algorithms that they can have a better readout, and also developing technologies will be much cheaper to work with. That, I think, will be a solution to what you're saying. And I think it's coming.
You know, there's several ways to approach it. One is double the NIH budget. That's probably the easiest. But there's actually, I think, organizational methods that could do. So, for instance, if TopMed allowed sequencing of libraries from sickle cell and we could apply for it, that reduces the cost of the assays by half, basically. Because when I look at it, and what will happen then is demand will reduce the cost of the technologies. So I think there are some things that administratively can be done. I agree that the cost is an issue in general. And, I panic that every now and then in my lab somebody wants to test transfection efficiency and they do single cell. I'm not happy with that. But in some ways, that's what we would like to do, because when you transfect a cell with a large non-coding RNA that you don't know what it's doing, actually there are different populations of transfected cells, and it's very interesting. But I think there are some simple administrative things that could be done to help drive the cost down, and I think the best program is actually the TopMed program. Because currently you have to apply with large population, but you cannot use single cell data for sequencing. And remember, 10,000 cells is like a good genomic epidemiology study.
So I have, there is a parallel from which we can import some lessons learned there, which is when RNA-seq was much more costly than it is today, and they needed standardization. And the same thing with the microarrays. When the FDA ran the MAQC and the CQC programs to exactly test the repeatability and reproducibility of these, if I recall correctly, the best was about 60-70% of the same sample across different labs or within the same core, multiple technicians, different instruments, what have you. So it doesn't matter what the size of the budget is. There are some limits in how much reproducibility, repeatability that you can actually achieve with any given technical method. So I would argue that we should look for reproducibility and repeatability at the level of convergent insights rather than the more raw form of the information itself. And that may be a bit more tractable and less expensive way to go at it. Because they've done this with RNA-seq and with microarrays, and they never got to 90% no matter how much money they threw at it, right? So there is a lesson to be learned there, I think.
A quick comment on essentially rigor reproducibility. Not everybody can handle that the way perhaps one should. And that's another part of big team science, to get the informaticians, statisticians to work with you and build a system so that you're much more comfortable with it after they do their math, and the biologists sort of nod and hope that they're correct. But I don't see that happening unless there's big groups of people that can handle it. That's a big challenge, and R&R isn't relaxation and all that anymore. It's rigor reproducibility that's coming.
I totally agree on that. In terms of really, the reproducibility after you have the data, it's easy. Bioinformatics, you can always go back, look at what kind of parameter sets you have for different software. So that's trackable. What is intrackable is during the process of generating those data, that is really hard. If you have the same technician, for whatever reason, maybe the shuttle to pick up the sample is slower for 5 minutes, whatever. So it could affect the results of your single-cell RNA-seq data tremendously. So I feel like that is really challenging part, the upfront data generation, reproducibility, to generate the data in a rigorous way, systematic way. It's very important. For that, it really takes a village. I mean, before I generated my own data, I thought data is just there, so easy. But after I generated a cohort myself, I realized you really have to, like she showed, you really have to get everybody in that party involved and informed, so there's no time delay. You really need to have a SOP in place to ensure that all the procedures are carried through the same way, exactly the same way. And statisticians definitely help.
I would just add that I don't think we need to be so despondent about this. It's okay to be doing early development of techniques, and I think commercialization will play a great role in this. So Salim talked about a lot of technologies that have been commercialized and as a result are more broadly applicable. So most of us showed data that was taken by PhD scientists, whereas we've all used a mass spec core or an imaging core where a technician can get the same result every time. So as the technology improves, I think there's lots of room for hope.
Mel Anderson from Cytovation. So I've been doing toxicology for 48 years. You can feel bad for me. I would say that the questions haven't changed. And I'd say when I listen to these tools, have the questions changed? I don't think necessarily they've changed, but there are new ways to look at the biology. And for me, the question that I ask is from toxicology, it's how you go from what we call normal, which I agree is a diffuse landscape, to somewhere else. So we're going to take a system and start to provide a perturbation, and at some point say the perturbation is excessive. For medicine, we're going to start with disease states frequently and try to go back and say what caused them and when was it sufficient. And in this regard, this idea of the Waddington's landscape and places where cells go, where cells are stuck for a period and then move. And I guess the question, the two people I heard, I think, talk about a little bit was Dr. Kaminski and Dr. Vadigepalli. There's this idea of what's a perturbation in a network. And I guess I'd like to know, from your point of view, did you see activity here helping you to understand how you go from one set of these cells, the multicolored cells, to another attractor state where the cell colors change and the numbers change? And with it, you can associate either disease processes or perturbations of normal biology that would call toxicology.
So one is the answer is yes, right? So, but it's based on old biology, right? So there's a set of things that I know. I know what TGF-beta does. I know what oxidative stress does. I find the same principles that I found before, it's just I find it in a different level, right? So now I'm not looking at a fibroblast on plastic. I'm seeing actually the evidence of joint effects of stressors on multiple cells. What we have not done yet is exactly so. Now take this model of the type 1, type 2 cell macrophage fibroblasts, the interactome between them, and now look at it in the fibrotic lung, and then try to understand what are the common perturbations. Because in some ways, it's actually, I've been been thinking about it the last few months since I started looking at it. So, is this a disease? The disease idea that starts with one cell going crazy, Or is this a disease that starts because the environment of the cell changes? Like if your cell starts secreting extracellular matrix, suddenly everybody's working in a stiff matrix instead of this, and they all change, right? Now we need a little bit more models, but I think now that we see the extent of changes that are happening, I think we could start differentiating. So if you think about, whatever, asbestos in makeup, right? You could actually try to make a model whether it's meaningful or not based on these systems, right?
So it's a fascinating question to think about the plasticity of cells as they traverse an abstract landscape of functional states, and what it means from trying to understand, answer the old questions that remain the same, if you will. For me, we're trying to find new, possibly correct answers at the moment. That might change later when we know more. And the new way of answering the old questions is thinking about what are the relative propensities of state transitions one way versus another, and which of those are controlling. And so of the many possibilities, which ones make it through the rest of the variation to affect the phenotypic outcome, if you will, in a measurable, substantive way. And if so, what governs those state transitions? It would be specific response to specific signals, or is it an intrinsic susceptibility of a particular cell type in a particular state? Or is it access to certain metabolic aspects? Or is it being positioned in a particular spatial location in the tissue that changes how a cell would behave? And we have seen examples of all of them. We have seen examples in the brain in the context of physiological perturbations changing the landscape itself and forcing the cells to move from one valley to another because the landscape itself is different. And we have seen examples in the liver where the landscape, at least within a time period of the regeneration, remains more or less the same, and the cells are traversing them because a certain signaling pathways are probably getting activated in a defined sequence, if you will. And so you can find both those examples. It's about mapping it back to a certain regulatory layer that you can then go test in an independent way.
And also asking questions. I mean, I think the way you showed the three areas in Statistico, that I'd never heard before. I want to remember that.
I looked for it. Google said it doesn't exist, so I just made it up.
So
that's what statistics we do, right?
I'll attribute it to you because I'll be using it within days, I'm sure.
In Simulo and in Statistico,
so
that's how I think.
It does seem interesting that when I hear you talk, the question is, how much of it is kind of a hypothesis-based and then bringing the pieces together? And how much of it today do you feel, because of these new tools and the data sets, are applying them to two legs of the stool, perhaps, and then creating an understanding that says you need to go out and use another line of evidence or of work?
I find that personally the dichotomy of discovery versus hypothesis-driven to be rather not helpful. Because at least in my practice, we tend to do both. I really want to think of it more like a what if. And some of that what if is really driven by, I have no idea, so I have to figure that piece out. And some of the what if is, what if this specific thing happens? And so I like to really look at it as a set of questions that are driven by what ifs and what may be, rather than which one is it testing a one specific idea where everything else is either controlled or known or in some way. But that's how I like to look at things.
So I'll just make a comment about this, which is, so I used to be extremely empiricist. My approach was, I know biology, but when I see something, I try to forget what I know about it and design experiments to test it. So when you do screening. What happened now actually with the single cell data is I find myself needing more and more to think about basic actually concepts of biology. So are these cells next to each other? Is there a reason for them to be? So the hypothesis is not necessarily X leading to Y, but the hypothesis that the fact that in a steady state, because that's a steady state, we find several things happening together, there must be a reason for it not to disappear. And I think that's sort of more of a, I don't know if you call it systems biology in a not computational, but just thinking about it.
Yes.
And that's what we've been going much more to these sort of frameworks and diagrams.
Part of it is the hypothesis is that we thought we knew more than we did, and these new tools are really giving us a different way to look at the problems we have. Thank you.
So I got a lot to learn, but I think, to me, some of the really important things that I'm taking away from this is that the variability that we often talk about when you get more to the tissue level or the organismal level is driven by what you're reporting on kind of at the cell-to-cell level often. And that I think we need to be kind of really understanding how to bound, or what the bounds of this are. And I love the comment, and Naftali, I think you were the one who said to it, about outliers. Maybe a couple of you did, but the point is they're telling us something really important, that instead of just saying, well, we're going to throw it out because it's, it's outside 2 standard errors or something, we really better look at it because that's what's going to give us some information. So I'm kind of looking at some of the questions that are up here, and especially 2 and 3, taking us to not only to the tissue organ and organism level, but I'm even thinking about taking it to population levels. And populations, whether you're talking about cells or you're talking about people or animals, in fact, instead of ignoring the extremes, we better pay attention to try to understand what's really going on. I don't know, it's just kind of more comment than a question, but I'd love to hear if there are any responses.
Well, I love what you just said, and if I might add to that, the main thing I would say is that the outliers are extremely important because the cell and the body are so good at buffering. So there's these limits where it's still normal and it's still okay, and eventually, when we were talking about these time points, even if you're out a little bit, you'll still come back. But figuring out the extrema is very important.
Well, I think the other thing is what is normal for one cell, one tissue, one organism, one person is not normal for someone else. And so I think we need to begin to kind of rethink our whole definition of what's normal.
As my original training in chemical engineering, I can't but help to note that there is an entire field in the process systems that focuses on outliers because that's where the learning to not make sure your refinery doesn't blow up actually happens, right? So the whole fault diagnosis and tracking, there's the whole field that totally contextualizing the outliers, I think, is the key. And having the high-dimensional, high-resolution information of the kind we have now provides that, to some extent, unbiased context to be able to get some value out of the outlier. Hence the reason they become useful. Otherwise, they're outliers otherwise. That's all you could say about them, right? So that context matters in that sense.
Yeah, actually, if we could learn from social sciences, so the context of positive outliers is actually established. So the people who are able to get food at times of hunger or not get disease when they live in a polluted area actually teach us about ways to protect yourself. Right, so I think that there may be an analogy there, and again, I think in the context again of environmental sciences, either the lack of response or the exaggerated response are fascinating. But now we have the resolution to actually measure it and characterize it, just not say. Yeah, so that's the context.
Basically, in a complex system, the concept goes like this. In a complex system, when you have adaptation and compensation, when that works very well, and like I guess tomorrow we'll talk about this ultra-sensitive systems and so on, you're bound to see outliers because there are cases where the system takes care of things. Our system is hypersusceptible one way or another. And so the outliers are to be expected, and there is a good reason why, because of the underlying regulatory system that's playing in there.
And finally, going from the last comment, going from a cell to a population, the outliers are important, as we know, because we can still identify vulnerable population or resistant population to certain conditions. So that will especially at the cell level, will probably tell us mechanism of action that we don't even know.
All right. That's been a terrific panel discussion here. I want to thank all of our session 2 speakers and all our audience members for the questions. We're going to move to a break here. We, of course, are running a little bit behind. Our break is supposed to end at 3:15, so if everybody can try to be back in at 3:15 or just a couple minutes thereafter, we'll get started as quickly as we can. A couple minutes before. Well...